So welcome back, sir. We were waiting for you to join and grace the dais. So, sir, are you there?
Yeah,
Yeah, he's
I'm
here.
there.
Yes, he's here.
Make me host, please. So I can
Yes, I can see him.
Yeah. Hi, Dr. Deepika. How are you? Lovely talk as always, and just enjoyed your being here. Thank you so much for
Thank
it.
you. Thank you for the invitation.
You got very clear concepts, the way what you gave is really very relevant. So,
That I think is 37 years of being a teacher, undergraduate,
and absolutely.
undergraduate, postgraduate.
And all Buddhu students like us, we learn from you.
No,
We learn
you
from
were
you.
one of the stalwarts.
Thank you so much, Deepika. So I'm
Thank
done,
you.
guys. You can allow me to share my presentation, and it's on epigenetics. I'm going to speak now for around.
Give us a moment. I think no, you don't need any introduction, but
No, I
still,
don't.
we want to say a few words. Dr. Meenu, please go ahead.
Ma'am, sir, of course, is right now currently heading his own iSeat, iHoma fertility clinics and the iSeat Training Academy, big time. He's a dean with Sridhar University Department of Clinical Embryology. He's president IFS. Got so many publications to his credit. He has taught so many students over the past 4 years, and we have no words to actually keep on. He's just the teacher of teachers. We owe so much to learning from him. He is extremely kind with his mentees, and he's a leader who walks the talk, and the CV says it all, and so the stage is yours.
Thank you so much.
Actually,
Thank you so much for the kind words. And now I need, Meenu, you have to stop sharing screen.
you need to stop sharing. So,
Please
Yeah,
stop sharing.
I'll go ahead. I won't keep you away from your chai pakoras in the evening because unfortunately I can't offer you nice chaat papdi in the evening now because we are online, but I'll allow you to move out earlier to have good time with your friends and your spouses. And here I am. Absolutely. So my topic for now is something very different and abstract. I'm basically going to talk about genetics and epigenetics. I threw in the word aging because I think everybody was speaking about PGS, PGT, NIPT. Dr. Deepika spoke so well about so many things coming around. So I thought I just catch up a new topic to create something fresh. So I'm talking about aging and how we are till now talking only about how we have an embryo getting affected by some sort of genetic defect. But now I'm going to talk about how we are getting affected by the genetic defects in totality. So we all want to look younger, but that doesn't happen because we all go in a clock. It's a big circle of life. You have to go through it. And rightly said, it is pretty enjoyable. And I think we all age very elegantly. Why is the topic important for you people? What do you know? What do you know? What do you all have to know about the topic? So look at these two pictures. You'll find that she's a very well-known girl. We all know about her. How gracefully she's evolving. And she's one girl who declined Botox. She said after around 10, 15 shots of Botox, she won't take it. She wants to age gracefully. And here we have our Brad Pitt. We all know about him. But overall, we all want to do something to look younger and look better. Now, what is happening is there's a big imbalance coming in the health equity nowadays because the women are getting married late. The issue is that they get married late and as a result, they conceive late. Or they may not conceive at all because something has gone wrong in their body. And if you look at this beautiful chart, you find that as the women are aging, the first thing to age, we also age, the first thing to age in the woman is the ovaries. First thing to go is the ovaries, as you can see here. And they are the one which create maximum chaos in her life. And in case you look at everything else, you'll find that her nerve conduction, heart rate, kidneys, liver, spleen, lungs, they age much later. So women, they age later and the men age earlier. Women live longer, men go earlier. The same thing happened with COVID. Now I'm going to apply the whole concept of genetics, that why does it happen? I'm going to talk about that. Few answers I will have. few I won't have. Then in case I don't have, I can call Dr. Deepika. She's there to throw light upon the concept which I present to you today. So can we do something to crunch our age? कोई ऐसा तरीका है जिसके थ्रू we can make our age shrink, I mean, to get expanded and we look younger and healthier. We're talking about having healthier embryos, healthier babies, but how about our body? Can we make our body look younger and more beautiful, more glowy? That's the way we all are designed to be. We all should be laughing all the time, but I don't see smiles anywhere. People are tired, they are stressed out, they're working around the clock and they're not looking good. We find many of our young friends, they have left us early. And why are they leaving us early? They've got a very good physique, they eat well, their environment is absolutely okay, they are rich people, and somehow they leave us. So why? What is happening to them, and what can happen to us also in case we don't take care of us? So there's a clock. The clock is clicking, ticking, and the clock is leading to low variant reserve. And as the previous speakers have spoken, Dr. Deepika spoke about it a couple of times, and so is Dr. Santiago. He speaks about it. They spoke about the X becoming weaker, more chance of aneuploidy, then we go into NIPTs and multiple testings, quadruple markers, dual markers. We start the whole process to find out whether a baby is healthy or not. And that's primarily occurring at a particular age. Before that, things are okay. So the number of eggs may fall down, the quality of eggs may fall down, the male will have their own problem, set of problems, sperm count may come down. So now when we look at it, when you're talking about genetics today and epigenetics today, let's look at it that, is it happening to me too? Are we aging? Am I aging earlier than my parents? There's one question which comes to our mind again and again. Since morning, we have spoken nearly for 8 hours. I think 8:15 was the time when I gave my inaugural address. Since then, time is now nearly touching 3. We have spoken non-stop with a half-an-hour break about genetics, how it affects the embryos and the human body. I think we all be on the left side of the picture. We all want to look bright and young. Aging is normal. We have to age. And aging is all controlled by our genetics. This is what we always feel. कि इसके genes अच्छे हैं. He's got better genes. He's from a royal family. They will look longer. But that's not the way the life treats you. We have seen many of the kings go early. They go early because of, not due to their pedigree, but due to the environment. And that is epigenetics. Genes can be carried forward, but epigenetics can be treated and curable. It is a reversible changes in the body which we create. So now, gentlemen, ladies, you have in front of you a beautiful chart. On the top side, we have is normal aging here, as you can see here. And below, we have got accelerated aging. And this is the chronological age. The chrono age is the age which is given on the passport. The passport says we are born on so-and-so date. That is our age. What is biological age? That means the age which we look like I may be 80 years of age, but I look 50, or I may be 40, I may look 60. So I've got an accelerated aging. So that's the way, this is visual. What we are seeing is somebody may look younger than their age, somebody may look not so younger as their age. But why does it happen? Why does it happen? Is it only genetics? We find that people from Northeast, the people from Nepal, people from China, South Asian countries, they always look, go to the Northeast, you'll find people are so young. Dr. Deepika will agree with me that the people in their side, they look very young. They never age. They always look glowing skin and younger and thinner. So what are the positive influences which affect their being young? One is everyday activity, proper sleep, nutrition, relaxation. And in case we have a lot of stress, smoking, lack of sleep, junk food, will accelerate our aging. So we may be 40, but we'll be looking 55. That's the way aging occurs. So this is, I'm going to play something for you.
They all require energy to function. This energy is created within our cells, in the mitochondria. Here, food is converted into chemical energy called ATP.
Am I audible to all?
ATP is released by the mitochondria, so cells can use it. Mitochondria consist of 2 membranes, an outer membrane, separating it from the cytosol, and an inner membrane surrounding the so-called matrix. The area between these membranes is called the intermembrane space. ATP is generated at the inner membrane of mitochondria by an efficient mechanism called oxidative phosphorylation, involving several membrane protein complexes. Nutrients provide high-energy electrons in the form of NADH, which are used by the protein complexes to pump protons from the matrix to the intermembrane space. This continuous pumping creates a proton gradient, where the positively charged protons are attracted to the more negative matrix. When the protons re-enter the matrix through the ATP synthase protein complex, they catalyze the production of ATP.
So what we learned here is something very, very fundamental and basic. When we eat our food, that gives us energy. That provide electrons and they go to the mitochondria. In the mitochondria, CoQ is the one which will further process the electrons and it will convert it into ATP. And ATP is the one which will give us energy. So we eat food, we get electrons, we have CoQ, then we get ATPs, and ATP will run our body. What is happening is that nowadays, due to certain factors, we are generating electrons, but we don't have CoQs because as we are aging, after 30-35 years of age, the levels of CoQ or ubiquinone in the body will go down. As a result, the electrons will start accumulating there. So guys, it's something like when you're on the cross-section of a road, you're driving on the road, there's a traffic jam. You'll find a lot of cars coming there. And then what happens when the cars come there? Normally cars are like electrons, they're moving very quickly. But when the cars get stuck at a point, they are generating fumes, they are honking, and they are creating a lot of noise and pollution. Same thing happens in our mitochondria. The mitochondria doesn't have CoQ, the electrons, they start multiplying, but they have nowhere to go. They start generating ROS, and that is the one which makes us age. So we have to look at the way, how can we direct our electrons by giving them more CoQ in case required, and getting the whole ball rolling again. That's what I'm going to talk to you about in some time. One part is about mitochondria. The mitochondria is a very important part of our body, and which we are ignoring. So whatever happens to us is due to mitochondrial aging. The mitochondrial aging will lead to all these things in front of it. So we have many terms. I've been listening to previous speakers. They've spoken about genomic instability. There can be stem cell senescence, mitochondrial dysfunction, which I'm talking to you about now. We have got deregulated nutrient sensing. This is happening very commonly nowadays. We've got proteostasis, which just the thing which actually Dr. Yukti brought out. He wanted to know about proteostasis from Dr. Santiago. Then we have got epigenetic alterations, telomere attritions, and genomic instability. So I'm going to look at this part of it and primarily focus on these three things. Let's see where I reach, and I'll be throwing in mitochondrial dysfunction wherever needed. The background of aging. Aging doesn't mean that we want to remove our freckles. Aging doesn't mean that I should start growing black hairs. Aging also means that we should have better quality life, we should have better quality ovaries, sperms, testes. So now let's look at it. How does the world behave? We have got something called aging. We'll define aging first of all. We'll talk about, are there any clocks by which we come to know that are we aging? So we have our calendars, we have got our date of birth. Date of birth will tell us our age. Are there any clocks which will tell us how we are aging besides our passport and Aadhaar card? What is reproductive aging and infertility? Epigenetics and genetics. This is the point which I'm going to highlight with a lot of love and affection. I'm going to talk about this thing at great length with all of you. And can we reverse aging? Can we do something to have age less? So coming to age and aging, the aging can be chronological. That is the Aadhaar card aging. Then biological aging is the one way we look like, how young we look, how old we look. Psychological aging. Some people may look younger, but they are psychologically sick. They feel that they have got their hypochondriacs. They feel they are infected. Then there is reproductive aging, where the uterus may not work well, ovaries may start aging. And then very important is sexual aging. This is my term which I brought out now, because I find that large number of men and women the common problem around 60-70% of them, they are aging sexually very badly. And I don't really have any answer for them that why is it happening to so many of the world people around us, besides our routine aging. So I've told you that we have two types of agings. We like to always have biological aging lesser than the chronological aging. So person should look younger to their age, and we have to focus on reducing our visual age. So when a person who's 50 years of age looks 30, he'll have a longer health span. Somebody who's 35 looks 40, he'll have shorter health span. This we can understand. Now, looking at understanding some new terms, the three terms we have: lifespan, life expectancy, and health span. So we are focusing on how we can have longer lifespan, which is healthy. This is what we are trying to focus on. So now coming to genetics. Now we have got aging clocks. There are two types of clocks which the God has. When we have taken birth, we got two types of clocks in our body. One is genetics and other is epigenetics. Epigenetics is a clock which is run by the environment. So what is genetics? Genetics is basically DNA sequence, what we get from our chromosomes and DNA, which regulates our genes. That is called genetics. And genetic will run from family to family. But on the other hand, we have got a major thing called environment. And environment means that something which will regulate this gene transcription. Here, DNA is not altered. In genetical problems, the DNA is altered. The DNA has some problems. But in epigenetics or environment, the DNA remains intact. But the DNA doesn't know how to work properly. So it doesn't transcribe properly, or the DNA can have the type of messenger RNA coding is occurring, that doesn't occur properly, but DNA remains intact. So epigenes are the ones which are sitting over the DNA. So for example, suppose this is a chromatin thread we have, or which is made up of various nucleotides. And on the top, this will keep, by pressing the button here, it will keep transcribing. But there are certain things which are sitting on top of it, which will regulate this. So the chromosome may be okay, the DNA may be okay, but the buttons being pressed are wrong. And this wrong button pressing is due to the environment. And that is called epigenetics. Now, epigenetics controls the working of the chromatid or the DNA. It doesn't cause any problem in the DNA. That remains okay, but it is not moving ahead properly. So look at these three things. These three things have not been covered to you since morning, and I think it was in my bucket. I had to talk to you about these three things. Always remember, there are three things we have to look at when we want to know about genetics. One is histone modification. Second is chromatin remodeling. Then we have got telomere shortening. Then we have got long RNA, long micro RNA modification, DNA methylation, and histone acetylation. These 4 or 5 things are the ones which are very important for all of us. We keep talking about epigenetics, but somehow we're not able to put epigenetics in the right perspective. As you can see here, we have got this DNA, double helical DNA. It is forming nucleosomes, and then the nucleosomes are getting together like a beads. It gets together, and finally we'll have a nice chromatin meshwork, and we have got the whole chromosome forming, and here we have the telomere. The telomere are the markers of our aging. So in case our telomeres are intact, they are longer, then we are doing good. But in case the telomeres are affected, they become shorter, there is a problem. There are two types of clocks we're going to talk about today. One is DNA methylation clock, and then is epigenetic clock. So clocks are not the ones which I have made or anyone has made. There are many types of clocks are in the market. One is called Horvath clock, other is called Hanu's clock, and they are available all over the world. In our country, we don't have it. But giving a sample of saliva, we come to know how are we aging biologically and how old we are destined to be as per our date of birth. So they draw a comparison and they give us our biological age, and by that we are able to do the correction. So we have learnt about NIPT, we have learnt about learnt about non-invasive PGS, invasive PGS. We had multiple speakers coming all over the world speaking about it. But about the human body, when we spoke about the embryos, we have spoken about the human body. We'll speak about it. Look at this. We know about your biological age through epigenetics. It's a kit called My DNA Age. Then you've got a kit like Atrinium. So they will tell you your age, your potential, and they all work on this particular principle. By focusing on some sites which are given here on the DNA, we have got some sites which get methylated. I'll be talking to you about them. We have DNA methylation. We have got histone unmethylation. And when we are able to see it by NGS, by taking a sample of saliva or blood, we exactly come to know what is the status of our epigenes in our body. And by looking at that, we can actually come to know that what are the accelerating risk factors for our body which can cause more harm, which are deaccelerating factors. So it's a certain factors which are good for us, which can inhibit a lot of things. And something like, I'll bring your attention to this. Let's not go up, but look at this. In case these are the beneficial lifestyles, and in case we get these things together, we sleep well, we have good income, good education, it's going to help us quite a lot. So this is about the basic principles of aging. Causes of aging, as you know, that it can be environment. Environment will affect epigenetics. The DNA person is born with, our chromatin will behave the way God has actually given us a DNA where we have taken birth. But biological aging can occur earlier due to the environment activating certain epigenes, and that is the leading causes are environment, lifestyle, drugs. They can all lead to early aging and aging of eggs and sperms. This is what is very, very important. So what causes accelerating biological aging?
The main reason why we age is due to the persistent DNA damage which takes place 1,000 times each day of our lives in every cell in our body due to the accumulation of the oxidative stress and also due to the environmental exposure which constantly expose ourselves to radiation.
So this is it. As I've told you, when there is a stress may be due to any reason. It can lead to generation of ROS, and ROS will finally lead to cellular damage. And that's all due to the epigenetics. Now moving ahead, looking at the various pathways, genetic instability may occur, and that may even be transmitted. A bad environment can accelerate aging because it can affect epigenes, and this is how it can affect the whole body. So environment is a very important factor which can lead to damage in the body, damage in our genital tract, and then whatever we do, PGS, PGT, we carry out genetic testings. This can't be undone. So we have to try to treat them young, catch them young. Now this is New Delhi on 1st of January. That's the way it looks like. And this is shores of any country. A lot of microwave using some sort of cream, phthalates, cream, sprays can all lead to aging. We may look younger now, but later on we'll have wrinkles using the food which has been artificially ripened, using acetylene, ethylene can lead to the similar problems. Powders to mature the food will make us age. So we are talking about organic farming nowadays. I guess we spray DDT over our food that lead to the biological magnification, and we keep getting more amount of DDT and chlorophylls in our body will lead to aging. And one big problem is smoking. So smoking is one of the very common factors. Smoking not only kills, it also kills our fertility. Will take away our dream of having the fertility, and smoking is, and this is a type of lifestyle we are having. A lot of fun, good fun we are having, but these all things will lead to multiple issues. And binge eating, I'm very fond of binge eating. I love eating all food. Minu is fully aware of that. So once we eat such a food, we are harming our epigenes. Mobile phones, telephony, stress, we are all aware, they all lead to, they are bad environment for our genes. Our chromosomes may be okay, DNA may be okay, but once we do all these things, our epigenes are affected. So now look at it, that reproductive aging will follow biological aging. In case we biologically age earlier, we'll also age reproductively. So all these things, HPO excess, and you look at this, that infertility, depression, osteoporosis, they all come together. Because the age is accelerated, all these things come together. Now look at it, the maternal age and aneuploidy. 81% of the eggs are aneuploid at 42 years of age. Look at this number. And it's primarily due to aging of the cells, but they are aging earlier in some women due to environment. And this is what I'll be talking to you for next 15 minutes now. Look at the chromosomal alteration. DNA methylation, mitochondrial DNA mutation, histone modification, non-coding RNA regulation. These are the ones which are genetic factors. And they are not only genetics, they are epigenetic factors which come into the play and lead to early aging. And all these environmental factors, they lead to these things. This is what we have to understand. We correct our environment and we'll correct everything. What we normally do is that we want to correct our aging by taking pills. We don't bother for that the incident cause. And the incident cause here is not the defect in the DNA, but the defect we are having, the defect is coming in the form of environment. So we have to correct our habits. You see that it's not only the female, but the male, when he gets married late, he will have decreased sperm production, quality and DNA integrity will go down. And there are few papers that the men married late, the child may have autism. There will be high mutation rate. That's the way things move on. And now I come to my main topic for the day. Gentlemen, ladies, I'm going to talk about epigenetics and mitochondria. We have discussed that we have to have good epigenetics along with the DNA. And when we have negative epigenetic age acceleration, then we live healthier. But in case we have positive epigenetics, that's a bad environment, bad way of living, then we age very quickly and become on the wheelchair. Some people live longer, some people age earlier. And here I like to say that genetics we already know. When talking about things which can control it, what all things can control it? And why does it happen? What is epigenetics? It's got basically three parts. They have histone acetylation, histone methylation, non-coding RNA action, and telomere shortening. All the things will lead to telomere shortening. Let's look at them one by one. So all of you are present here. We have around 94, 95 of you are here. Just take a snapshot of this picture. We have non-coding RNAs, which is a very important part because they don't code properly and they are the ones which are actually having some defect in coding. They're called non-coding. They are called siRNAs. Then we have got DNA methylation and histone modification. There are three things which I'll be covering for you again and again for next 10-12 minutes. Look at the slide properly. We have two things coming in our DNA. One is protein, other is chromatin. So remember that proteins are always having positive charge. They are histones have positive charge, and chromatin has got negative charge. So based upon that, I'll take you further in this journey. So we have got histone which can be defective. There can be DNA which can get defected due to epigenetics. And then there is other thing is we have non-coding RNA. So all these three things can occur due to bad environment, and they can give rise to aneuploidies. So this morning we have planned the meeting in such a way that we're always talking about how to correct, when to biopsy, when to do NGS, NIPT, because we are getting poor quality eggs. But in case we improve our epigenetics, that thing may not be required at all because we'll have better quality eggs. A woman of 35 or 38 or 40 may look like a woman of 30 in case we do things properly. Now, as we are young and as we age, then the diseases, they start increasing in number. This is all which we find occurring in the body. There is chromosome and chromatin metabolism goes very high. We can have wrong transcription of the DNA, and there is non-coding RNA maybe there which can lead to problems. And there can be defective RNA, defective histones, defective chromatin network. There can be defective or wrong production of proteins. And finally, we have high amount of proteolysis and housekeeping genes, they may become defective. And I'm going to talk about these things as I move forward. There are two terms. One is called methylation and acetylation. These two terms are very, very important. And just understand that we have got a molecule of cytosine. The cytosine, the fifth place, this is number 5, terminal. We are getting an methyl molecule. Methyl molecule is taken by DNMTs from SAM and it goes and gets stuck here. So when a methyl molecules move to cytosine molecule, nucleotide at the 5th position, we say that it has got methylated. And all of you who may be wondering what this methylation is, I tell you one thing, that methylation drives our life. In case we can control our methylation, we are all done with our life. So please understand the concept of methylation. Moving off methyl molecule by DNMTs from SAM molecule, that is S-adenosine mononucleotide to number 5 position on cytosine, that will make the molecule will become, the DNA will become methylated. And methylation is important. Look at this top on which we have put the thread around, say, Nattu kehte in north. I don't know what you call all over the world. But in case we have this, suppose this is a DNA we have, chromatin, and this we have with the histone protein, they are strongly bonded with each other. And as I told you, that our histone proteins are always positively charged and DNA is negatively charged. So in case they are binding each other well, that is due to the positive and negative charge. And in case something goes wrong, then the whole thread will open up. And when it opens up, we say that it is going defective. It can open up when the time comes, but in case it opens up automatically at wrong times, it becomes defective. And just remember that a DNA, when DNA is lying open like this, it is called euchromatin. When DNA is bound, it is called heterochromatin. So we always look for a balance of euchromatin and heterochromatin that we look at. And when it opens up, so can someone tell me what do we have here? It will be euchromatin or heterochromatin. Anyone? It's you or hetero? Anybody?
Euchromatin.
Euchromatin. Fantastic. Euchromatin. So now how does it happen? It happens very beautifully due to this algorithm. We find that we have got, these are the histone proteins, and there are 8 of them. You see, 1, 2, 3, 4, 5, 6, 7, and 8 of them are there, in which we have 2 circles of chromatin coming. So 8, 2. This is called, it will lead to formation of nucleosome. And this is nucleosome which is forming. And to fix the chromatin over the histone, we have got pinning. And this pin, like we have to put a pin there to fix this thing, the chromatin on the histone. And that is done by methylation. If you look here, it is called DNA is condensed. And what we are having here is methylation. You can see a proper methylation, then the DNA is properly bound around the histones, and we have got very good DNA. And that is called methylation, and methylation is good. In case we get acetylation, it means that this thread will open up. The thread will open up, and so will be the bonding of chromatin with this. It is called acetylation, and acetylation is bad. It should occur in a balance, but when you start aging, then methylation stops occurring and acetylation increases. So when it increases, the whole of chromatin will come free and chromatin will start transcripting, and that is not a good thing to have. So we have to have a balance of acetylation and methylation. Methylation means is good, and acetylation is also good, but it should be done in a controlled way. So loosely packed acetylation will lead to formation of euchromatin, and methylation will lead to formation of heterochromatin. This is what you have to understand. Something goes wrong, our telomeres in the end of chromosomes will get defective. Another picture. I wanted to go back with a very clear answer about these things, what occurs in the human body, so that you start catching the people earlier and do proper counseling about their lifestyles. Now, let's look at it. Here we have the DNA. As you see here, the DNA is making nucleosomes like this, and finally it forms a chromosome. Chromosomes will have telomere in the end. So whatever happens, the telomere will get shortened in the end, and we start aging. So we have to stop telomere shortening. And look at this. Here we have, in this case, we have got, due to methylation, we have got condensed chromatin. And when we have got acetylation, we have got open chromatin. So as you can see here, when we have got methylation of cytosines, when we have methylation of cytosines and deacetylation of histones, then we have got gene is off. Gene is not working. So your chromosome chromatin is safe. But when the gene is on, it becomes open, it will become euchromatin, it will become unmethylated, and histones will become acetylated. Acetylation means that the charge between the histones and the chromatin, which is positive and negative, that goes off. So they will start separating now. Like 2 magnets, they hold on to each other. We take 2 magnets which are holding to each other. Like suppose I have in my hand I have 2 pens, they just hold on to each other. It's like histone and chromatin coming together. But in case something happens and then they separate, then they will separate and fall off. It's called acetylation. So we are looking for methylation and deacetylation. This is a basic concept of having good quality eggs, good quality embryos, and healthy aging. Look at something different. There's something called CpG islands. In the audience, we have got more than 95 people here. Can someone tell me what are CPG islands? Anyone? Does anyone know the meaning of CPG islands and why they are important? Anyone can make an attempt. We have heard about CPG islands. Can anyone make a guess? No guesses. The CPG island, this island where we have some amount of, it is cytosine phosphate guanine islands, where we get these sort of CPG nucleotides coming together, and they are basically called the housekeeping genes or the promoter genes. Now, they should always be active. So CPG genes should be active and they control the RNA and DNA transcription. They're very important genes for us. So they should always be active, and the moment they get a stimulus, they should start transcripting. They are the islands. When they occur together in a group of 100, 200, 250, they're called CPG islands. And they are the promoter or housekeeping genes. On the other hand, look at the whole DNA. The whole DNA has got different molecules, and when these molecules are methylated, we are happy. But the CPG genes should be unmethylated. So I hope you're getting my point. In a young patient, look here, in a young patient, the whole of chromatin is methylated, so it is suppressed. But CPG island is unmethylated, so it is working. It is working. But as we age old or as we become cancerous, that time we'll have global hypomethylation here. So look at this. You've got all the methylation will keep reducing, but here, look at here, CPG islands start becoming methylated. So they stop working. And as a result, cancer occurs and we start aging. Is it clear to all of you that the basic concept of all the problems in our body is that when the CPG islands start getting methylated and all other chromatin meshwork, it becomes unmethylated. So CPG islands the balance of CPG islands with the other parts of the DNA nucleotides is very, very important. And this is what I just want to tell you, that there is something called age-specific epigenetic landscape, which is common. We have to age. It's absolutely okay. But we have locus-specific hypermethylation, and we have global hypomethylation as we start aging. And global hypomethylation is a marker of cancer, marker of our aging after 40 years of age. But along with this, what happens is that there is hypermethylation of the CPG island. So that leads to, that's a marker. That's the way we can take a drop of our saliva and check the CPG islands and find out how many of them are getting complicated. The CPG islands are getting stuck, they're getting DNA methylated. You know that this person is aging or he's got a disease in his body. Then we can compare the other patterns of the DNA inheritance, methylation, and we can give somebody an age in the difference of 2 to 3 years. So then there is altered histone modifications. When the histones, they become, I've told you that they become acetylated. When they become acetylated, the histones will open up, they lose their charge, and the chromatin will fly off. And as a result, it'll start transcripting and the DNA will become elder. Look at this lovely small video I'm showing for you. Here you find the nucleus in between, and outside is you can find there is some mitochondria, lysosomes. This is what is happening in the body, in the nucleus. You'll find that there is RNA strands, DNA strands, which are bound around the histone proteins. The magenta ones are the histone proteins. They are the histone proteins. And here we have got the genes being transcribed. And this is euchromatin. And what we have here is the heterochromatin. And finally, when the genes are being transcribed, they come from nuclear gateways. They come out. They come out in the cytoplasm. In the cytoplasm, they start making proteins. And these proteins are the ones which run our body. In case something goes wrong with this whole assembly, things get messier, and something is controlled here. So in case the right genes are being suppressed, things won't happen and there will be problems. So this is basically an DNA stand. As I tell everyone a lot of times, that one human body has got DNA of nearly 2 meters. And you bring the whole DNA together, you can go around the sun 3 to 4 times. So this is the purple one is the histone proteins. Outside is the chromatin. When there are 2 loops around 8 histone proteins, we get a nucleosome. Then finally, the solenoid, when they come together, nucleosomes will come together and form the DNA. That's the way the whole thing occurs, and that's the way you can see the chromosome is being formed, and this chromosome, these DNAs, chromosome will keep multiplying and dividing as the time is required, and then we have the pattern of 46XX or 46XY in the male and female. And this is they are the killing machine, it's a factory, it keeps making proteins. Whenever they open up, it becomes new chromatin. When they're closed, that is chromatin. Everything works on the basis of acetylation. Methylation will stop it, and demethylation will open up. Acetylation will again make it new chromatin, and they start multiplying. When it occurs very, very rapidly, then it can lead to damage in the body and start aging. Telomere shortening is an important thing, and this is in case we have a chromosome. Think this is a chromosome, and the terminal end of the chromosome is called telomere. And when telomere has got a particular length, when it becomes shorter, then we say that the person is aging. Look at this man who's 40, maybe around 70, 80 years of age. He may be around 50 years of age, a child. Find that the telomeres are getting shorter. So telomere shortening is the key to longevity. This we can measure by certain techniques. And again, come back to the same chart which I started earlier. Look at how the things work in the body. And our main aim should be to avoid telomere instability. And for that, we go to avoiding telomere attrition. Then there is something called SIRT gene, that is silent information regulator protein. That is a protein in the body which is very, very good for us. But when the amount of SIRT in the body will become low, it's called a sirtuin gene. It's called SIRT127. One is important for us. When the SIRT falls down, we start aging very quickly. So we want to increase sirtuins in the body. High, then we are doing good. Levels of SIRT are falling down, we start aging, and it can directly affect the aging, and we have quick, rapid aging. And what does SIRT they do? SIRT will lead to methylation, and it will lead to deacetylation. So the same thing which is spoken about, the more we can keep the DNA intact together, it is called methylation. And the more we can avoid acetylation, it is a good thing for us. And the sirtuins can do this by using by using the molecule like NMNs. It is very effective, and it is also now available in the market by some generic names. Then the second molecule which is important for us to avoid epigenetic changes is called NRF2 gene. Now this gene is found in the cytoplasm, then it goes in the nucleus and it makes the endogenous antioxidant gene. So that's a very, very important part of our body. In case we have high levels of sirtuins and NRF2 genes, then we are doing good. The options we have is primarily good sleep and diet. In case we sleep for around 8 hours in the day, we have proper diet, we can definitely do well. That's very, very important for all of us. They will all lead to reduced environmental stress and better genomic stability. So these are the molecules: ergothioneine, resveratrol, metformin, rapamycin, CoQs. So I'll just take you through in 2 minutes through them. We have got something called NMNs, that is called nicotinamide mononucleotide. This is SIRT activator. So when SIRT increases, the whole body becomes better. Multiple papers are there over the SIRT proteins, and it is an antioxidant. It is a genomic stabilizer. NMN is a good drug which can be taken, and here we'll find that everything which we are thinking will become better. Telomere integrity will become better, and then these are few papers over NMNs. So this is very good for eggs. In case we have high levels of SIRT, eggs are of better quality. Second is ergothioneine. It is NRF2 activator, again available in the market. It's a natural antioxidant. So very good at the level of molecular level. We'll have better outcome. We have better type of blastocyst embryos, and we may not require PGs and PGT for them. Another drug is called rapamycin. It is called the sirolimus, and it comes from an island called Rapamui, and comes from a bacteria, and it also acts on an mTOR pathway. And finally, whatever it does, it will reduce the epigenetic changes and making it is a transcription initiation factor. So it will make good quality proteins, and the eggs will be of better quality. Human body will be of better quality. So lifespan will be increased. In case we take rapamycin, some books have written, but I'll leave it here. And second drug is called the resveratrol. It is found in grapes, and the grapes, you can take them raw. Some people take wine. If you look at the French people or the Japanese people, they age slowly, because probably they take a lot of grapes and red wine. So it's a very good antioxidant, and it is anti-aging. It acts at the level of epigenes. It's a very good drug. One good drug is metformin. Metformin, 500 milligram per day, can actually lead us to better lives. It can delay aging. And again, it is acting at the mTOR pathway, AMPK activation, autophagy activation. So we can have extended longevity and lifespan. And finally, they all lead to telomere lengthening. All these drugs will avoid genomic instability. They will reduce cellular senescence, reduce inflammation. So all these drugs will have healthy aging and longevity by acting at the level of epigenes. Diet, I always say that we should take, in case we take intermittent diet, is very good for us. And whatever all the drugs will do for us, the diet will do for us. They can correct the methylation defects, the acetylation defects, all can be covered by taking proper diet. And all that, or everything comes together, and intermittent fasting is as effective as taking any molecule. It's good for us. CoQ10 acts at the level of mitochondria. I've spoken about DNA, epigenetics. Mitochondria, I've told you already that mitochondria are the most important thing in our body, because they affect the spindles directly. Because the spindles become weaker, then they cannot, the chromatins cannot separate, they become aged, they lead to aggregation and improper segregation. And there comes the concept of PGS and aneuploidy screening, NIPT. In case we can boost our old eggs, and we can boost our eggs oocyte sperms, that condition may not occur. We will get good quality eggs and sperms and embryos. And CoQ10 is a very important molecule. As I told you that in the body, the levels of CoQ start falling down after 40 years of age. Some book says that 35 years of age. So in case we do supplementation with CoQ, we can have better outcome. The CoQ supplementation will enhance our mitochondria, enhance our cytoplasm, and we can live better and longer. Maybe we'll have more normal embryos on PGS, less of mosaics and less of aneuploids. So disruption of the CoQ10 pathway leads to meiotic outcomes, bad meiotic outcomes. This is from Robert F. Casper. We all know about him. It leads to proper DNA assembly, proper as you can look at the spindle. It is a good quality spindle in case we use CoQs. The OCD complexes are better quality. So here we'll have less chance of aneuploidy. Look at this. Since morning, what you have learned is how do we detect it. What I'm telling you is how do we correct it. That's by the method of using epigenetics and environment. So there are large number of papers that we take. In case we take CoQ, we can have better outcome in terms of better quality oocytes. And there are multiple papers from ASRM and all over the world. People are saying that to reduce aneuploidy and mosaicism, they are using CoQs, and they get 62% reduction in the aneuploidy. So I think there's one thing which I leave it at you. Now, how do we, like we do NIPT and NGS, what do we do for aging and looking at epigenetics? So there is a concept of AI, where we can take some samples from our body, and then we come to know what type of the island of cytosine phosphate CPG islands are getting affected. We can look how is the methylation and deacetylation occurring. We can draw the ratios from the normality from the people who have more biological life, and then we can develop some deep biomarkers. We use some artificial intelligence. So these are things which are available all over the world. I mean, all the big countries probably will come to our country very soon. So to conclude, I would like to just end here by telling you that when we talk about aging, we all want to become younger, but we can't become younger by using PGS and PGT and PGM. We'll become younger by improving our environment and epigenetics. This is a topic I wanted to keep for you in the beginning, but unfortunately I got stuck in the morning. This should have come in the early morning session. So green environment is the one which will give us better quality oocytes and better quality embryos. Yes, definitely yes. PGT, PGM, genetics we cannot. DNA is one part of it, but what controls the DNA is again a very important part of our journey toward getting a healthy baby. Good quality diet, good quality drugs you can take in case required. So, but the final endpoint is that we have to cure our telomeres. In case the telomeres are of this type, good telomeres, this can only come due to epigenetics. So what we are doing is we are detecting bad telomeres and then doing NGS and NIPT and multiple things. But in case we strengthen our DNA, make our chromosomes and telomeres stronger, then probably the requirement of that thing may come down. So a good diet is the one which we hate. जो भी दुनिया में चीज अच्छी है खाने के लिए, we don't like it because that doesn't have that spice in it. All the good food we don't take. We all like chaat papdi and masalas. So definitely we have to face the world. If we take healthy diet, stress, various applications, sitting for 10 minutes a day and internalizing will probably strengthen our chromosomes much more than later on going for NPT and those sort of things. Relax, being with your wives and with your families can make a lot of difference. Yes, saying no to smoking is very, very important. And in case you want, such kits are available in many countries. You can try it there. They will give look at it. Here, 42 is the biological age and 47 is the chronological age. So this person looks younger. It's 0.89 of your age. So it's a very good method. This person will look younger always. He'll live longer. That's the way they give you the outcomes. And I think just wanted to open. So thank you so much for being with me. I just wanted to open a new Pandora's box for all of you because I thought I'll take you to some different level. And you understand, we all keep talking about chromatin, DNAs, RNAs, but I took you to epigenetics. That was the aim for me today. Thank you so much for being with me. I think I've taken around 10 minutes more. So Nisha is very angry. I can see that.
Yes.
Very angry. But I wanted my pound of flesh, Nisha.
I didn't even know, sir, where that 10-minute went. I was so engrossed in the talk. It is really amazing, wonderful. Like this way, we never thought about eggs and sperms and embryo. We keep telling, I am sure that everybody keeps telling the patient to improve the lifestyle. But in these terms, maybe if we explain, patients are even more receptive to it and they will be able to understand. Wonderful talk, sir.
Thank you
Wonderful.
so much.
Wonderful.
Hi, Yogesh. How are you? Nisha, can we have a group picture with everyone now? Is it
Yes,
possible
sir.
for you to get everyone
Everybody
together?
can
I
switch
can see
on their.
good Yogesh, Meenu. Thank you, Meenu. Thank you, Yogesh. Thank you, Nisha, for putting it on. It wasn't an easy task for all of you. It took us around 2.5 months of preparation, and I think it's been a super hit. Holding
Can we
100,
have
100
everyone
people
to,
together,
can I request everyone to switch on their cameras, please?
keeping 100 people together for morning 8:15 to evening 4:15 is not easy, Nisha.
It
And
was, no, sir, it
both
was
Meenu,
wonderful. All the speakers are very, and the last talk is like very good, very good.
I think we had very good delegates today. We are not thanking them. I think we should
Yeah,
thank
we
them.
will thank them.
So, guys, we want
We
to
have
see
a
you.
picture.
We want to see you. We want to thank you for being with us. Hi, Shravani. I can
Hello,
see you
sir.
over here. कैसी है? How are you doing? Good to see you.
I joined Nagpur.
Okay, I know that. I keep chasing you everywhere. I know you're in Nagpur. I keep chasing you. Fantastic. Other guys,
I want to talk to you personally.
अभी फोन कर दियो फिर. अभी फोन कर. खट से फोन कर दे. अच्छा. So can we,
Everybody
guys,
please switch on their cameras. Can
Yeah,
we request everyone to switch
for
on
few
their
minutes,
cameras?
for few minutes. I think we want to thank you, guys. We want to thank you. Really want to thank you for being with us. I want to thank you personally.
Please switch on the cameras.
Sir, it was a very good session. And really it has cleared many of our doubts, which even we don't know that we are having that doubt in our mind. That basic conceptual training was very good.
Thank you.
Sir, we should have one get together at Delhi.
Delhi करेंगे. जल्दी करने वाले. We have a lot of plans. So I think I like to thank place my records.
Do we have a
Please
picture?
thank the records for Nisha and Meenu. Thank you so much for this. And third is Yogesh, who has been working very, very silently and regulating the whole program. Thank you, Yogesh. Sanjita, I can see Shravani, Anupama, Raksha, Priyanka. I just want to see you and thank you. That's all. Upma. Hi, Upma. How are you? Great. Arpita. Then I have Mamita, Santoshi, Divya. I have Anu. Hi, Anu. How are you? Thank you so much for being with us. We really enjoyed. Thank you so much. Great. Ajay, Ruchika, Purnima, Shweta. We have Bill Broad, Nikulu. Thank you so much. Divesh. We have Kovart Singh Parihar. Thank you. Sachin. Then we have got Ashwani. I just want to thank everyone and we want to really thank you from our heart, whomsoever I can see. Komal, Kajal, Sudhas, Anubha, Santosh, Vikas, Monica, Tushar. So thank you so much, guys, who have put their cameras on and we soon meet again. That's all I can say. So, but Professor Prakash is here. Dr. Baligar, are you there?
Yes, I'm listening.
Yeah, so I'd like to hear from both Yogesh and Professor Baligar in case we have time or anyone wants to speak.
Sir, it was a very nice lecture. Since morning, I am sitting here only to hear all the topics. They were very, very excellent and very knowledgeable.
Thank you so much for supporting us from MIT University. That was fantastic. So, Yogesh, anything from your side? You are an embryologist. No, we can't hear you, Yogesh. We can't hear you. You are muted. Anyone else from the audience would like to speak?
आवाज म्यूट करना
Can I?
Yeah,
Am I audible?
yeah, you're audible.
Yes. The way we started since morning, we are studying and learning and thinking about the DNA. Everywhere is DNA. So thinking, learning and DNA from zero to its role everywhere it is role. So at last, you spoke well about the aging and the DNA. So when we are going to take care of DNA, then we can take care of everything. And especially when we are working in the field of infertility, yes, this DNA is controlling everything. DNA is master molecule for me every time, and whatever the protein, the mRNA, the transcriptome, the genome, and the proteome, whatever we are handling, it is coming from the DNA. So getting such of the conferences and such of the webinar or the get together event. So they are boosting our knowledge and they are giving us some of the insight for this DNA. So such kind of events should be keep on going so that we can learn more and more. And thank you very much, sir, for having us with you. And every time that we are the part of this, and such a nice and very well done program. Thank you so much.
So I think we always thank the woman behind the camera or behind the screen or on the screen. I like to thank one man today. Where is Mohit? Is Mohit there?
Mohit is there, sir.
Mohit, can we
Good
see
evening,
on the
sir.
screen? Can you see on the screen?
Yes, sir.
On the screen. I can't see on the screen.
Am I visible to you, sir?
No, you're not visible. He's invisible. I can't. There's so many screens. I'm not able to see him. I can see all your
Yes,
lovely.
he's
Yeah,
here,
Mohit.
sir.
I can see Mohit now. So Mohit, it was fantastic. I think you deserve a big clap from all of us. This was not possible without you.
No,
Please
sir.
share from Mohit.
Sir,
Thank you so much, guys. And Tisha, back to you. And Meenu,
Yeah.
back to you.
So if anybody has any questions, I would like, this is the right time to ask now. Anybody has
your
any question?
session was very good, madam, very informative. We would like to have your presentations copy or literature, whatever you can do.
You can see them on our YouTube, madam. Dr. Shravali. Go on our YouTube.
Yeah, it will be available on YouTube. Okay.
Go to our YouTube channel and subscribe it. Subscribe it also. We'll put everything there. We'll put everything there, madam. Please
Yes,
go
I
ahead.
will
Don't
just.
talk.
Sir, I would like to say something.
Yes. Yes, Super. How are you?
It was such a heavy webinar since the morning, but your talk made it so light, like we're feeling so lightheaded. Oh my
Yeah,
God.
the topic was difficult to make it understand, but it was very easy.
Thank
It's
you.
very interesting. It was interesting, but it's a very heavy topic. But now, right now, we're feeling so lightheaded, like all the burden has been lifted off, and we have come back to the basics.
Yeah.
Like improve lifestyle and improve such things.
Or
Thank you, sir. Thank you so
how's
much.
your daughter? Where's your daughter? I'd like to say hello to her in case she's around.
Sir, my daughter, I want to say hello to my daughter. Oh my God, she's giving a test.
Topper hai wo. So before you see her on the
Sir, to hi,
big boards and ordered by
Yes, sir.
Modi ji. Hi, how are you?
Hi, I'm just giving an exam.
I know. Studying for NEET now. When is the exam?
Mock.
Mock. So
A
when will,
major test.
so when will
Major
Yogi ji
test.
give you an award? Very soon, Yogi ji will give you an award.
Uh-huh.
Very soon, very soon. God bless you.
Thank you, sir.
I think that's all from my side. And would anyone like to share anything? आपको बात करनी चाहिए, आपको बात करना चाहिए हम सबके साथ में. अगर आप करना चाहें तो हमें अच्छा लगेगा. All these videos will be there on our YouTube channel, I think in a day or two. But in case you want to speak to us now, we'll be very, very happy to take your queries. Anyone? All are tired, Mr.
No, sir. I have certain, I think there are many thank yous in the chat box. So many of them from Ritu, from Dr. Sachin, from Dr. Karanjit, from Raksha, from Leelavati, from Shrisha. I think everybody is like very clear about many things. You know, she's exactly, Upma is absolutely right. Since the morning, it was becoming, with each discussion, it was becoming like, more and more burden on our head के बाप रे कैसे पेशेंट कंसीव करेंगे इतना सारा है पीजीटीए
It
पीजीटी
was like what
कैसा
to do, what not to do, and
but
which test
yes
we will miss
NIPT
out.
and this and that. Now with one final talk, we are like, more or less clear that if they eat well, they exercise well, they rest well,
Yeah.
they do their own proper dieting and meditation, maybe there is some hope that they might not need all those things.
There are a few questions, Nisha. I just saw.
I want to ask one question, sir.
Yeah, please ask. Whatever I
Yeah.
know, I tell you.
Yeah. Sir, no, it's a very simple one. Actually, sometimes we find that people have missed the time for double marker and their level 1 lenti scan.
Yeah.
Now, if they're opting for a quadruple marker from 16 to 20 weeks, what is the right time to get an ultrasound done again? Because for an anomaly scan, they have to wait for.
I'll pass it
82
on to Dr. Deepika in case she's there. She's the fetal maternal guy. In case Deepika can't take it, then I'll pass it on to Nisha. Because I don't follow pregnancies till so long. I see till around 8 weeks. So Nisha, you can take it on, or
This
Deepika.
is happening quite frequently for the patients who are in villages or remote areas that they miss
Yes,
their
ma'am.
scan. So I would suggest that the best thing is we can do, even if they come at 14 weeks or 16 weeks, you should get one scan done so that you know, and even at 14 to 16 weeks, you can get a quadruple marker done. So in Gangaram earlier, we actually started with quadruple marker. This dual marker and all came late. So if the patient
triple marker was earlier, ma'am, I suppose.
So if
Earliest was triple
marker, yes. So
Yes,
if they
ma'am.
miss the scan, you can get it done at even 14 weeks, get a triple marker done, or at even 16 weeks. So I think whenever you get hold of them, you should get one scan basic done.
I think quadruple marker is the preferred one. Ma'am, triple is almost
Yes,
out
ma'am.
now.
Triple was started earlier. Now it is quadruple. Like when we started, it was triple marker.
No, my question was just one thing, ma'am, that quadruple marker, the timing is from 16 to 20 weeks. And say if you have to go for a level 2 scan now, what is the right time? Should we do it at 18 weeks and then plus like add on with quadruple marker? Or should we do it at 16 weeks only and then get your level 2 at 22 weeks?
So 14 to 16 weeks, you can do the scan and triple marker. 16 to 20 weeks, you can get the scan done and quadruple marker. 18 to 20 weeks is the level 2 scan. You cannot get the level 2 scan done before 18 weeks because that doesn't give you the exact.
Yes. No, yes. So basically, we should see that if triple, yeah. So ma'am, just last question. If you have done a triple marker because she has reached to you at around maybe 14 weeks, would you like to go again for a quadruple also? Because triple is not we are not doing a lot of triple marker nowadays. Either you prefer quadruple or double marker or go for NIPT.
I would prefer double marker. NIPT they are suggesting is a first line treatment, but no, I think each case needs to be individualized. You see the age of the patient, you see the scan. If a scan is very well done, accordingly you can advise the patient. No need, no problem, no point in repeating the test which is already done.
Yeah, okay, ma'am. Okay, thank you so much, ma'am.
So
It
Nisha
was a
should do one
wonderful
thing.
session.
So Nisha, you should do one thing. This is a very good question. About which many of us are not clear. IVF people, we follow till a particular time. We have to make a small algorithm of this and given the group. So that everyone should get a written protocol of this. This you do for us, right?
Yes, sir.
Within 24 hours. But so everyone knows what is happening.
I'll do that.
And everyone will benefit by that. So I think we have some questions about from Sanjita Pal. Is Sanjita there? Sanjita has put a query that can CoQ benefit male also. Am I right? That's what you wanted to know? Yeah, it can improve every quality, Sanchita. The reason being that it acts on, I told you, the mitochondria become defective because the CoQ is not there. With the aging, the ubiquinolons become less in the body. So when we are eating good food and ATPs are being formed, the ions are being formed. Ions are formed, but they are not being transported outside the body or outside that mitochondria. They start choking there, and this can lead to problem in the sperm as well as the egg or an embryo. And sperms have got they're very rich in mitochondria. The sperm neck or the mid piece is very rich in mitochondria. So it will affect the quality of sperms also. CoQs are very important for male. What supplementation is preserve reproductive age? I think that reproductive aging, that is as per our biological age, we have to protect our biological age in case we want to protect our aging. And supplementation, there are no supplementations per se. I say that in case you want to really do well, I always prefer that eat less. Anyone who's eating less will have everything is auto-corrected. So eating less and having 8 hours of sleep. रात को 8 घंटे सोना है अच्छे से. That's very, very important. I think everyone should buy a watch. I personally feel that everyone should have a watch like Fitbits or any watch, Apple Watch, and look at the balance of how many hours are you sleeping per day. You should be having good amount of REM sleep and non-REM sleep in a proper index. Sleeping for 8 hours is very, very important. You don't need any supplementation. If you want to take after around 30, 35 years of age, I think I tell people to take metformin. I advise large number of people to take metformin. 500 milligrams per day is a good drug. It's a very good drug. Metformin and intermittent fasting is very good for us. The people with whom I work all over the world, it's a group we have, they are prescribing 41 to 60 tablets a day for aging on average. They also go in to check in the male penile turbulence. They check the penile turbulence every week or 10 days. Like we put we halter to look at the penile turbulence also. That because how is the penile artery is aging. There's a lot of things are happening in the world. And I say that please, in case you're really keen to know about this answer, the right answer will be given to you by David Sinclair. If you get time, type David Sinclair, a lovely book by David Sinclair. Will tell you, will answer all your queries. And Weber role of resveratrol. Most doubtful resveratrol comes from, I say it comes from grapes and wine. If you want to take a tablet every day, you can take a tablet. It doesn't do magic, but definitely is an antioxidant. You can buy it, nothing wrong in it. It won't harm you, but it doesn't have any direct benefit as per the data which we have. And people can take it. I say have more good grapes and blueberries. They are good for that. And then Sanjita has said this. Thank you. That's all I think so. I think I'm done. Nothing for me. I can see that. Nothing for me, Nisha. I'm done.
So with this, I officially, we are at the end of the day, and it was a great day. I would like to extend my deepest appreciation to all our distinguished speakers. And, we had the speakers from all over the world. Their expertise, their insights, and their dedication have truly enriched our understanding. It was an awesome day with various, this field of genetics is very complex, and they have provided invaluable knowledge to all the attendees. I am very sure that all of the attendees have gained a lot. I also like to express my greatest gratitude to Dr. Pankaj Talwar sir, who's the mind behind it. He's been saying thanks to everyone, but he's the one who actually everybody needs to thank to, because he's the one who started this thing. He's the one who said that we need to do this, and he's the one who even pushed and encouraged me to do this. Thank you, sir. Thank you so much. And I think Meenu is the other one which I need to thank to. She is a very great support. She helped us all a lot. Our attendees, the people who have attended the whole day, they were here with us, listening to each and every talk. And the foremost thanks also goes to Mohit. His support is invaluable. I think n number of times I told him to make the creatives and change it. He always did it with a smile on his face. Mr. Vinod, Mr. From Progenesis, Mr. Baligar from MET, and Monica from SCRT. They all are also they did a very great task of helping us in concluding this workshop on not even the whole day, or in time also. So thank you so much. Thank you so much, everyone, for being here with us the whole day. And
even I would like to say a few words, ma'am. Of course, sir is the idea behind it, and he is, the guiding force. But ma'am, you have done a splendid job of coordinating it all for us. And of course, I must big mention to Mohit also. I have seen him evolve. He is a very hardworking person. Big time support to sir and the entire IC team. Thank you for the opportunity, sir, ma'am, and the entire IC team.
I am very happy that I am part of it.
Thank you, sir.
It's a passion only, I think, which can keep us glued the whole day on a Sunday with this genetics thing. Thank you, sir. Thank you.
Thank you. Bye. Take care, everyone.